Okay, with biolistics, what you've got is, does that get any bigger? No, it doesn't. Okay. You have a tube, a gas acceleration tube, and then you have this rupture disk which comes out, and we have this microcarrier. And on the microcarrier, what you have is DNA covering, DNA-coated microparticles, microprojectiles. So it's like a shotgun with really teeny, teeny, tiny balls. And the microprojectiles are so small they can enter a cell without killing it. The microprojectiles can be tungsten or they can be gold, and they are coated with DNA. So you have this machine with the gas coming down. It's a blast of helium that comes down, and it hits this microcarrier, and then suddenly the microcarrier flies down, and then it hits this stopping screen where it stops, and then the microcarrier stops, but the projectiles that are on the microcarrier continue, and they go down and they hit the target cells. So the way this was originally designed was, let's see if we have another picture of this. Yeah. So originally, this was designed by John Sanford to have you had a 22 shell up here, and that provided the energy. And you hit the 22 shell with no bullet on it, like a starter pistol, and that would explode, and this would move a cartridge all the way down here, and then the cartridge would fly down here, hit the stopping point, and then it would all spray down. But now they're using helium instead to accomplish this. And so here's an example of one and what it looks like. And then the cells that it hits can be anything. And the way it was originally done was you'd take a sterilized leaf and you would hit this and you'd blow the center of it and it looked like someone shot it with a shotgun. And then the inner part's useless because it's mush, but the outer ring, you could take those cells and cut them out and grow them in plant tissue culture, and you come up with transgenic plants. The great thing about this for transgenic plants is that no agrobacterium is involved at all. Agrobacterium works very well with some dicots, not all, but some dicots. But it works very poorly with monocots. And of course the monocots are the ones you're most interested in, maize and rice and wheat and barley, all those things, so it doesn't work very well with them. So when biolistics came along, they had a method they could use for that. And then they tried it on mammalian cells, and it worked really well with mammalian cell culture. So that was really great too. So then they finally commercialized it, and they came up with this Helios gene gun that you could buy from Biorad. And it's actually a handheld unit, and you can go ahead and work on cells, you can work on tissue, you can work on all sorts of things. So biolistics is really a neat way to do it. Okay, we get the DNA into the cell. What happens next? How does it get into the genome? It goes in the nucleus, and then what happens? The way we've seen with all these other technologies we've been looking at. What happens next?
Some phage recombinase?
No, not a phage recombinase. No, it happens naturally. Homologous recombination. Yeah, homologous recombination. With plants, it doesn't work that well, so we have to use non-homologous recombination. But that's how it gets in. But you can use these for all sorts of things, including DNA vaccination. So they're used in lots of different ways. Or you can use it on plants, right on the plant. Boom! Take that. Or you can use it in mammalian cell culture. So there are all sorts of ways you can do it. Okay, so that's a really fun thing to do. Well, next we're going to look at is chloroplast transformation. Oh, that doesn't look very good. Yes, that's what I want. Okay, so with chloroplast transformation, your first question is probably why, but the reason is the Scandinavian research. Okay, is this getting bigger? No, it doesn't. Okay, that's the biggest it gets. Well, anyway, the way this works is, well, maybe get a better picture. These are all small. Okay, maybe this would be bigger. No, a little bigger. Okay, that's pretty good. Okay, so here what we do is we are going to transform chloroplasts. Now, there are going to be about 100 to 1,000 chloroplasts per cell. Usually, no, it's about 100. I'm just thinking about when I look at cells. Yeah, maybe 100 or 200 per cell. And there are multiple genomes within chloroplasts on top of that. So the great thing about if you transform in chloroplasts is you have many more copies of the gene than if you did nuclear transformation. Nuclear transformation, how many copies of the gene are we going to have? Well, maximum 2, because we're diploid, but usually you only get 1 in. So you usually don't get a homozygous. You only get 1 in. So 1 copy of the gene, that's not very much. But with a chloroplast, if you have 100 chloroplasts and you've got like 10 copies of each genome in the chloroplast, then wow, suddenly we're up to 1,000, so that's 1,000 times better. So the great thing about chloroplast transformants is they produce at a very high level. Second thing is, what really are chloroplasts? Are they prokaryotes or are they eukaryotes? They're basically prokaryotes. So the great thing about prokaryotes is you're basically, if you have chloroplast transformed plant and you put it out in the field, you basically have a whole field of E. coli that you don't have to culture. You just provide dirt and water, and you've got a field of E. coli. And so they tend to handle genes, and prokaryotes tend to handle genes better than eukaryotes. They're less picky about what kind of gene you put in. So it's more robust that way. So for all these reasons, chloroplast transformation is fantastic. Another thing about chloroplast transformation is, okay, where do you get your mitochondria from? From your mom, from your dad, or from both?
Mom.
Only from mom. Why is that? Why don't you get mitochondria from your dad? Well, you know, it's no mitochondria in sperm. Okay, so it's the same thing with plants. When they have a gamete, the gametes which are produced in the pollen do not have chloroplasts. So if you think about it, if you had a plant that had problems with interbreeding, so for example, if you were growing sunflowers, for sunflower seed, they do a lot of that in France, for example. The problem with the transgenic sunflower is it's going to crossbreed with all the other sunflowers, especially if you do it in America, because sunflowers are native to North America. All the sunflowers are going to get that gene eventually, because the pollen from your sunflowers will be taken by bees to the wild sunflowers, and you're going to get introgression of the gene into the wild populations. Another example is with the Canadians, is they like to grow canola to make canola oil. But canola is basically a mustard. It's a brassica. And there are all sorts of other mustard weeds that are present in right around January, February. If you look in here in Texas, you'll see these golden plants on the hills. They're the only thing blooming. That's all mustard weeds. So if you plant transgenic canola, for sure it's going to get into all the mustard weeds. Now, other plants, like if you plant rice here, there's nothing like rice in North America. There's no wild natives. There's nothing like maize in North America excluding Mexico, just U.S. and Canada. There are no wild teosinte plants. So those kinds of plants, the pollen could go everywhere. It's not going to breed with anything, so it's perfectly safe. But there are some plants you have to worry about. If you have chloroplast transformation, the pollen, that transgene is going nowhere because there are no chloroplasts in the pollen. So that's another great thing about chloroplast transformation. How do you do it? Well, you go ahead and transform one chloroplast, and then you have to grow this on special medium. Let's say you gave to it a kanamycin resistance gene. Then you have to, if you were to give that antibiotic right away, you would kill the plant. You'd kill the chloroplasts because you only have one chloroplast in the whole plant. And so if you killed off all the other chloroplasts, the plant would die, right? So what you do is you slowly give the antibiotic, and then you have to grow it out, and then you step it up and add more, and step it up and add more. So months and months and months of this culturing, and finally you get a plant where the bulk of the chloroplasts are transformed. Because chloroplasts, you have to think about it, these are prokaryotic invaders in our cells, just like mitochondria, and you have to treat them that way. They are a population of organisms living inside our cells, and you have to slowly get them the population changed. And so that's the thing with chloroplasts. Okay, our next one of the journey here is we're going to look at some genes that we can put into plants, and we're going to look at 3 of them. The first one is from Bacillus thuringiensis, and we talked about this before. Now, Bacillus thuringiensis is related in a group of bacilli, which includes Bacillus cereus, and I always love Bacillus cereus because it's abbreviated B cereus, and then Bacillus anthracis, which causes anthrax. And in reality, if the taxonomists wanted to, they could make the whole group Bacillus anthracis, but then suddenly Bacillus thuringiensis would go up to biosafety level 3, maybe, which we don't want. So we're keeping it here. What is Bacillus thuringiensis? Well, there you go, right there. Dipel is something that is what the organic farmers, the San Francisco type people who want to not use pesticides and want to do something totally organic, and they buy dipel. And if you're going to be doing organic farming and you want to not have caterpillars rather than spray them, you buy dipel. And what that does is it's pure Bacillus thuringiensis Kerstachii. And the Bacillus thuringiensis have these subgroups. So Kerstachii is the subgroup for the ones that kills caterpillars. And you spray this stuff on your crop. The little caterpillars eat this stuff, and then they die. It's a bacterium. And there's another one, Bacillus thuringiensis israeliensis, and that's another version. And they sell them as products. And they sell these, you've probably seen these before, mosquito dunks. They sell them at Home Depot. They're little donuts. And you drop a mosquito dunk in your pond, and it's just a cake of Bacillus thuringiensis israeliensis. And it grows in your pond, and mosquitoes who take it up are killed. Or Bacillus thuringiensis, it gets rid of fungus gnats too, that's cool. Bacillus thuringiensis, because I've got that problem with that with my greenhouse. Bacillus thuringiensis israeliensis, what it does is it only kills mosquitoes, sandflies, tsetse flies, which are all vectors of human disease. So it's just fantastic. And this is the one we use in our lab. We're making transgenic plants for this, so we can express nectar, which expresses it, so we can kill mosquitoes. But anyway, so Bacillus thuringiensis is really a fantastic group. But we're going to focus on Kerstachii right now. And it is what this does, as you can see here. Let's just get bigger. Yes, great. Good. This is a good picture. So if you have BT corn or BT cotton, and we call it BT for Bacillus thuringiensis, these are transgenic plants. What normally happens, the reason the organic farmers love this bacteria is it produces a crystal. So you can see right here, this is an original TEM, a transmission electron micrograph, and that big thing right there is the spore, and then this thing right here, they colored it purple, but that is a crystal. But they really are, they're just that spectacular. They're gigantic things. And what happens is this bacterium is dead. It just has a spore and a crystal right there. The caterpillar comes and eats it because it's laying on the plant. And then what happens is it gets solubilized in the acidic gut of the bacterium, and then it becomes activated by proteases in the gut, and that breaks it down from the BT protein from a really big molecule to a small molecule. And then once it becomes activated, it binds on, as you can see here. Here's the midgut cells, and it binds to cadhedrans, And then there are other proteins as well that it binds to, but when it gets to the dihedrins, it forms an oligomer. And then it goes in here and it forms membrane insertion. This becomes a pore. And of course, when you have a gigantic pore lining all of your gut cells, then it's just like cholera to the 10th power, and all these caterpillar die very quickly. Then the spores, you see, because we had the crystals and we had the spores, then the spores are able to grow and activate, and then they come in and they have this caterpillar all to themselves, and that's how this organism lives, right? So, if it's these proteins that are doing the job, then you should be able to use them. And here's a picture right here, yeah. And so look at that crystal, isn't that gigantic? And here's another crystal right here, and then behind you the spore. So it's amazing, really great stuff. And of course, Israelensis works on mosquitoes. Yeah, more crystals. Here are some crystals right there too. And let's look at, we don't have any pictures of cryproteins. Well, we have different cryproteins, and each cryprotein, each one forms a different crystal. So those crystals you saw, one might be cry4a, another one might be cry4b. There are different crystals that these things form. So what they did then is they took the cryproteins from this organism that forms the crystals, and they made transgenic plants for them, and the plants are resistant to bacteria, to caterpillars. And they also have different cryproteins that confer resistance to beetles, like Colorado potato beetle. And so that's one of the big technologies that's used in plants today. And that's called Bt corn, Bt cotton, Bt soybean. That's just the abbreviation they use. Okay. So let's go on then to Roundup resistant crops. This is the next really big area that is used. And so here we have in USA, so with soybean, the percentage of cultivated land, with Roundup resistant soybean, this is just Roundup resistant soybean, this is the only trait here. You can see it's very popular with soybean, very popular with cotton, and now it's catching on with maize as well. And so, whoops, sorry. And they call it Roundup Ready. So we have Roundup Ready alfalfa now. They introduced Roundup Ready soybeans. And then this is really funny. See, you have all these activists which are, having all this bizarre, they're just very, very odd. Monsanto fails and all these things. They're just really weird people. Very strange. So, yeah, see, they have this sort of stuff. So they don't, it's just attack of the superweed. It's just the most bizarre thing in the world. I deal with all these sorts of things. So I deal with creationists an awful lot. Because like in the church I go to, oh golly, I would say 90% of the people are creationists, something like that. And so, being someone who's done evolutionary work and published in it, the one thing about the activists that I can say, and also with the creationists, is in general they have good hearts. They want to see something good. And the creationists, what it really is they want to see the Bible preserved. And they feel if somehow creationism is knocked down and we interweave evolution with Christianity, that Christianity will fall apart.
And it doesn't. In actual fact, Christianity becomes far, far stronger when you weave in evolution and you weave in biology. And I could go into that in detail, but it really, Christianity as a paradigm really supports evolution far better than any other faith I've seen, including secularism. It really weaves together really nicely. Secularism, if you mix secularism with evolution, you get some really scary stuff that goes over to Sartre and Nietzsche and other things. But if you mix it with Christianity, it really comes out with a lot of integrity. It works quite well. You have to look at Genesis 1 through 11 as allegory, but that is very common for that Jewish writing. That's the only shift you really have to make. But in general, creationists have good hearts. In general, these activists have good hearts too. They really fear that something terrible is going to happen to the world if we go all genetically engineered plants. Of course, we are all genetically engineered already in America, and nothing terrible has happened. There have been no health hazards, no ecological damage. But I think you just kind of get caught up in the whole thing. So that's how that works. Anyway, so with Roundup resistance, what we have here is Roundup, which is a compound, is fantastic. When I was at Cornell, Roundup was just coming out, and in my soil microbiology class we learned about it, and our professor was so excited about it because the other herbicides cause a lot of environmental damage. Roundup is really benign, and it degrades within 2 weeks. It's a modified amino acid, and so it was very exciting to get that. But basically what we have here is we have, here's with tobacco, is you have the disc removed, and we're growing these discs out. You have callus, and then you have a shoot, and you transfer over. And just using the same procedure, we're now putting the Roundup resistance gene from bacteria or petunia into this. And now the plants are going to be, you can go ahead and spray the plants and the weeds together in the field, and you wind up with crops that are completely unaffected, whereas the weeds all die. So that's basically all it is to Roundup resistance. So we can move on from there and go to virus-resistant transgenic plants, another huge success. Whoa, what did I do? Well, virus-resistant papaya, we could start with that one. And this is from my home lab, from the lab I got my PhD in. And this was done by Dennis Gonzalez, my supervisor at Cornell. And this was the first resistant plant marketed, and it was done by a professor. And it was the last one done by a professor. After that, they made it so expensive to do all the ecological tests that they couldn't afford it anymore. But basically the way this works is that you have, so here is a papaya plant which has papaya ring spot virus, and here is a papaya plant which is transgenic. And papaya ring spot virus can really be devastating. As you can see, these plants, they're really destroyed.